microtubule dynamics. The next two lectures, we will now spend more time to look at microtubule TIP tracking proteins, and I will start to give you a bit a view what kind of mechanisms are involved for these tracking mechanisms. But before I come to that, I very briefly want to give you a little overview what my group or what our lab is doing actually. So we are interested in protein. No, it's not.
Hold it a little bit closer to your mouth.
Closer?
Yeah.
Like this? Like a singer? Rock star. So, our group is interested in protein interactions that regulate the microtubule cytoskeleton, and I don't have to give you a reason why the microtubule is so interesting, because microtubules are involved in many fundamental biological processes. One of the most prominent one, perhaps cell division. And because microtubules are involved in so many fundamental biological processes, you also find several diseases that are directly linked to malfunction of the microtubule cytoskeleton, like cancer. Again, one very prominent example, but also, as we have seen during the workshop, also neurodegenerative diseases. So, protein interactions regulate the microtubule dynamics. Since we are living in a systems biology area, people generate these very complex interactomic schemes, right? And these people in 2010, they have mapped all the interaction that either directly or indirectly act on the microtubule cytoskeleton. And it turned out that there are more than 300 proteins that do that. And I think it's here the secret is encoded why the microtubule cytoskeleton actually can participate in such diverse fundamental biological processes, starting from cell division to intracellular transport, etc. The reason is hidden somewhere in this very complex network. And that's more or less our playground, where we try to pick up specific questions. You cannot target all the 300 proteins simultaneously, and the approach we use is an integrated structural, biochemical, and cell biological approach. And most of the time we really try to produce high-resolution structural information, because my belief is that in a structure there is much information encoded that you then can exploit to learn something about what happens in the cell, and I will show you two examples on that. We also use classical biophysical, biochemical assays to characterize our proteins, and ultimately we really want to go into cells to test hypotheses that emerge from the in vitro work directly into cells. And this part of the work I always do in collaboration. One fantastic collaboration I have since a few years is with Anna. So we have done a lot of work together, and we managed to go from cell biology observation to in vitro observation back forth and stimulate so the progress of the research. So we really aim at producing structural and quantitative information of the system to get detailed mechanistic insights into the processes. So the projects that are ongoing in the lab are centered around the microtubule cytoskeleton. We have a project geared for analyzing tubulin-modifying enzymes. Peter Bass has told us a bit about them. We also look at tubulin-targeting agents, because my belief is that learning from these agents, you also learn something about the microtubule system itself. We look at molecular motors. We also recently looked at centriole proteins involved in central duplication, but quite a lot of work in the recent year was focused on these microtubule plus-end tracking proteins. Okay, so this movie Anna already showed you. What are plus tips? Plus tips, they have the capacity to recognize and localize and track the growing microtubule heads. And that's a situation where we have transfected this so-called EBH-SXIP protein that is fused to GFP. In red you have the microtubules, in green the EB protein. And it turned out that quite a large variety of different proteins, they show this localization behavior just as I showed you in the movie. And when you look at these molecules, what sticks out immediately that they are really unrelated in structure. They are maybe multi-domain proteins, yes, and they sometimes share domains, but otherwise they're completely unrelated. They can be relatively small, like the EB proteins, roughly 300 amino acids, but they can be as extensive as 9,000 residue in a row in the case of the microtubule acting cross-linking factor MACF. They can be membrane bound or even integral membrane proteins like the STIM1 family members. We have older proteins that show this tracking behavior and Eclipse and P150 group that were introduced already by Anna. Now, interestingly, they are not only unrelated in structure, but they are also unrelated in function. So these plus tips, they don't exert always the same function, but in principle you can find all types of functions. Starting from regulating microtubule dynamics, that's probably the most prominent property, function property of plus tips. So they can be polymerases, we already heard a bit about that, rescue factors, catastrophe factors, or depolymerizing enzymes. And besides that, they can also mediate interactions of the growing microtubule end to cellular structures like the cortex, because they are very well positioned to be the first molecules that touch an object. So they are very well situated to mediate these interactions, or with the endoplasmic reticulum, or the crosstalk between microtubules and the actin filament system, or also mediating the contact between the microtubule tip and the kinetochore chromosomes, or microtubule crosstalk. Another properties of these plus tips is that, so besides they are unrelated in structure and in function, they form tight interaction networks. And actually they are very dynamic interaction networks. So when you look at, so these are individual proteins just showing also the conservation across evolution. They are tightly interconnected with each other, either by direct or indirect interactions. And it turned out that the master regulator of these dynamic plastic networks are the members of the EBs. And I will give you now two reasons why not only we, but other people think that EBs are indeed master regulators of these dynamic plastic networks. The first reason is that the EBs can autonomously track growing microtubule ends. And these are, if you look at the other cases, most of them, they don't autonomously track. There are a few examples, but EB is the most prominent. How has this been shown? That's a paper. We will discuss more extensively this afternoon, but I just give you a glimpse what it was about. So what these people did here in 2007, they used GMP CPP stabilized microtubule seeds. They immobilized these onto a glass surface. They added tubulin subunits, GTP, and the end-binding protein. And by doing so, we're able to reconstitute the localization very reminiscent to what you see in cells. So this is a purely in vitro system. You only have tubulin, only GTP, GMP, CPP, stabilized seeds, and the end-binding protein. And you see very nicely, if you follow this microtubule here, it decorates the growing microtubule, and while one microtubule shrinks, this comet-like appearance disappears. So exactly what you observe in cells. So that's a good reason to believe that EB is a master. It can do the job by itself. The second good reason to believe that EBs are master is that it turned out that EBs can directly interact with a huge variety, if not with all other plastics that are known so far. And it turned out that these other factors, they rely on the direct interaction with EB to localize to the growing microtubule. So, two good reasons to believe that EBs are master. Now, in this slide, I have summarized more or less the current knowledge, how EBs look like. I told you they are not too big, 300 amino acids roughly in length, but they form dimers, and dimerization is brought about a coil domain in the C-terminal part of the EB. The N-terminal part of the molecule has a cloponin homologic fold, typically found in actin-binding proteins, but in this case, it's this part of the molecule that mediates the interaction with microtubules, in particular with the growing microtubule, and we will again hear this often by the students, how the CH domain manages to recognize only the growing microtubule N and how it distinguishes it from the rest of the lattice. I don't go more into the details. The N-terminal domain is separated from the C-terminal dimerization domain by a long linker, roughly 70 amino acids, predicted to be floppy. We don't know too much about its structural behavior. Then, as I said, we have a coil domain, and we also have here at the end a 4 helix bundle and a disordered tail that terminates into an EEY F motif. And I will come also back later what is the relevance of this EEY motif. And it's this part that I boxed here that is called the EB homology domain. It's as a sequence domain and also translated into a structural domain that is unique to EB proteins. You don't find this domain in any other proteins. And interestingly, it's exactly this part of the molecule that recruits other plastibs. And I will also come back later how this domain actually manages to recruit other plastibs. Now, the question that we had said a while ago is, I mean, how can that be? How can that be that such a small molecule is able to interact with these completely unrelated proteins, and sometimes even very big proteins? I told you up to 9,000 residue. How is this done? So the story I want to tell you is from this paper that is again a very strong, as I told you, a strong collaboration done with Anna, and it's Srinivas and Susanna and Anke, Srinivas and Anke from my lab. They were PhD students, and Susanna, PhD students in Anna's lab that carried out all the work that I will show you. So the first thing, when we looked a bit about the information that was present in the literature, we realized, so here you have the EBs, we realized that all these proteins here, they use basic serine-rich domains predicted to be intrinsically disordered to bind to the C-terminal part of the EB. So that's an information we had from the literature. But if you looked at the sequences, they were not conserved. So how can you make sense out of this? And what we did, we used a bioinformatics approach. We used an algorithm called DILIMOT that was published by people at EMBL in 2005. So what does DILIMOT? DILIMOT identifies short 3 to 10 residue linear motifs within a set of non-redundant proteins sharing a common structural, a functional property, like, for example, a binding partner. So these molecules here, they are unrelated in function, in structure, so not members of the same protein family. However, they share a common function. The common function is they bind to EB. So that's exactly what you need to input into this algorithm. So what we did, we created a data set of 12 non-redundant plastid sequences, always one representative of each family, so no redundancy. And they had to contain basic and serine-rich domains, and from the literature we knew they bind to the C-terminal domain of EBs. And what we then got from doing this is that there was a small motif, serine, X, isoleucine, and proline, that was found to be the highest scoring motif candidate within the set of these 12 proteins, but not only this, also cross-evolution. So a very strong signal in these sequences that was carried on from yeast to man, a 4 amino acid motif. And you can appreciate this motif in this sequence alignment. Here you have a few examples of plastids that have these basic serine-rich domains. In the middle, I have highlighted this SxIP motif. And indeed, when you look on the left and on the right side, there is hardly any other conservation. Maybe perhaps beside that we see an enrichment of lysine and arginine residues. And I will also come back what is the role of this lysine and arginine residue. But otherwise, that's the feature we have seen. So obviously, that's something that was very exciting to see. So obviously the next question, before we have spent more work on that, was, well, is this something relevant what we have found? Is this a motif that is now functionally relevant? And the best way to test this is to go directly into cells. So what we did is to pick a few plus TIP examples and started to investigate them. The first one is MCAP. Don't have to introduce you what MCAP is. As we already heard, there is an N-terminal domain, a central motor domain, and a C-terminal coil domain. And as a dimer, it's a 160 kilodalton. It's 160 kilodalton in size. There is a basic serine-rich stretch right here with an SxIP motif. So what we did is we fused the molecule to GFP, transfected into cell, the wild type situation, and compared its localization behavior with a mutant where the isoleucine and the proline were simultaneously mutated to asparagine. And what we got is that while the wild type, as expected from the literature, very nicely TIP tracks growing microtubule, the mutant where we just replaced isoleucine proline to asparagine completely failed to do so. And at the same time, also in pull-down experiments, it was only the wild-type MCAP molecule that bound to EEs while the mutant failed to do so. So we were able to already draw here a direct correlation between EB binding and localization to the growing microtubule tip, and that these two residues are absolutely essential to do that. The next candidate protein we looked at many. I just show you basically two examples where the clasps, so the clip, associated proteins. Again, we have a basic serine-rich region in the middle of the protein, and interestingly, in this case, there are two SxIP motifs that popped up during our sequence analysis. And what we did is now we tested whether both are functionally important by doing a similar approach what we did with MCAP. So first we looked, so we only looked at the middle part of the molecule, not the full length. We looked at that wild-type fragment, compared it with a mutant where the first IP motif was mutated to asparagine, then the second IP motif was mutated to asparagine, and then we compared those to the double mutant where we mutated both motifs. And as you can see now, or appreciate from these time-lapse movies, the middle part of this fragment behaved very nicely. TIP track growing microtubule. The two single mutations, SxIP mutation, you see that TIP tracking is already reduced, while the double mutant was completely dead in the sense it did not localize. So it's showing us that proteins can also accumulate multiple SxIP motifs, either in tandem, as we have seen it for clasp, or when you have coil domains that oligomerize at the class tip, you can also get multiple motifs. And this having multiple motifs makes the process more robust. So important to keep in mind, these motifs can occur in multiple copies. Then, based on this, we ask the question, well, what is the minimum requirement you have to add to a GFP such that a GFP molecule can localize to the growing microtubule length? And for this, we used a fragment from this huge protein called MACNF, microtubule cross-linking factor. Again, it C-terminals as a basic serine-rich region, one very nice SxIP motif, and it turned out that you, in principle, only need these 12 residues fused to GFP, and you get the localization behavior as if you would have added the full-length protein. So demonstrating that that's the secret of these molecules to bind to EB and to track growing microtubule lengths. Now, during the course of the work, we also wondered, well, can this interaction be regulated? Because in biology, many things are regulated, and we were fortunate that at that time there were quite good papers telling that in MCAP there are 5 Aurora B phosphorylation sites. And phosphorylation is typically used for regulation, and when we looked where these 5 Aurora B phosphorylation sites clustered, 3 of them, or now even 4 of them clustered in the vicinity of this SxIP motif. So to test whether phosphorylation has an impact on MCAP localization, what we did is we again compared the wild-type situation with a mutant in which we replaced 3 of these 4 serines to glutamic acid to mimic phosphorylation. Okay, glutamic acid is negatively charged, a phosphate group is also negatively charged. And what we found is that indeed, by having this serine replaced to glutamic acid, localization was strongly reduced. I mean, you see maybe a little bit, but compared to the wild type, there is really a big difference. And the same is also true in protein assays. The phosphomimetic mutant failed to interact with EBs. So, what is now the structural basis? What is the mechanism of these SxIP motifs? And structural biology is a very good tool to exactly answer this question. So remember, that's again the EE. We have the N-terminal calponic homology domains for microtubule binding. We have the C-terminal domain harboring this EE homology domain. So what we succeeded is to crystallize this part of the molecule in complex with a peptide harboring an SxIP motif. And the first thing we saw in the structure is that this floppy C-terminal tail, this 20 residue, parts of it get structured. You see this part here? That's normally in solution is disordered. It gets structured. By doing so, creates a very nice cavity to accommodate these 4 SxIP residues. And so it turned out that, so we have a flexible, I mean, this peptide is a round coiled solution. It gets ordered upon binding to its cavity, but at the same time, also this part which is flexible also gets ordered. So there is a neutral induced fit binding mechanism involved in building up this complex structure. So when you look at in more details about the interaction, we saw that the isoleucine of the SxIP motif sticks into a hydrophobic cavity. We see that the serine is engaged, making hydrogen bonds, and also the proline is deeply inserted into the cavity to contribute to the final affinity. And when you look now at the sequence conservation of the interactions that are made between the EB homology domain and the peptide, in red are the residues that we have seen that contact the SxIP motif. You see that they are conserved down to yeast. So actually they're not only conserved, they're invariant basically. So telling us in the end that EB homology domains are SxIP regulation domains. That's their function. Their function is an across evolution to recognize these SxIP motifs. Now, I told you that phosphorylation can regulate the activity. Can we explain that based on our structural information? Can we also get an idea why phosphorylation tunes the interaction? Again, we can look at our structures and check for the charge distribution of the EB homology domain. So again, here you have the EB homology domain. Here you have the SxIP peptide. In red, I plotted the negatively charged residues, and in blue, the positively charged residues on the surface of the EB homology domain. And you can immediately recognize that the charge, the surface charge, is biased to negatively charged residues. Now, I told you also in the beginning that what we also saw is that the flanking region of this SxIP motif is typically enriched in positively charged residue like arginine and lysine. And actually, when you look where they locate in the structure, they are very close to these negatively charged patches. So, telling us that there are probably long-range, unspecific, long-range electrostatic interaction that stabilize the complex. So we have two aspects. We have something that generates specificity, that's the SxIP motif, that fits like a key and a lock into the pocket. And we have long-range electrostatic interactions that add to the stability. And now what we think, since the phosphorylation sites are here in the vicinity, they will just interfere, because they are negatively charged, with the negatively charged surface of the EB homology domain. So that's what we think how phosphorylation works in this context. Now, based on all this information, the last experiment we did at that time was, can we now convert a complex protein that is not a plus tip, can we convert it to a plus tip? And for this, we again went back to the NCAP system, because NCAP is a kinesin 13 family member, or KIF2, depending on the old and newer nomenclature. And this family contains two other KIFs, namely KIF2A and KIF2B. And then there is KIF2C or NCAP. They all have the same domain organization. they all have this basic serine-rich domain in the N-terminus. However, only NCAP has an SxIP motif, while KIF2A and KIF2B, we were not able to see any SxIP motif. So, predicting that these two guys should not localize as wild type to the growing microtubule end, and that's exactly what we saw. So, NCAP, this you have already seen, beautiful localization. These two other members that do not have SxIP motif, they do not localize. So, we use now KIF2A to engineer, just with a few sequence changes, a canonical SxIP motif in this region, and we were able to transform this phenotype into a phenotype where we change immediately the molecule into a true plus tip. And that's actually the secret, or no, that's the strength of these linear motifs. You can acquire with just a very few sequence changes a dramatic change in activity, and that's the strength, because they can mutate or evolve much faster as if you have to build a full domain. And there is a lot of additional work on linear motifs that in principle show exactly this kind of property. So you can acquire an activity with very little sequence changes. So based on all this, we have proposed a concept that SxIP motifs are general microtubule TIP localization signals, or MTLS, in analogy to nuclear localization signals. I mean, we did not invent a localization signal. There are other localization signals that have been reported much earlier, but all use the basic principle I just showed you. A linear motif, you stick it to a protein, and if it has a receptor, you can localize it to the right location. Are there questions concerning this part? Yes?
So, if you bind the protein or if you receptor, that is not like a microtubule polymerization, because it's very long, when you don't have the domain.
I think Anna will show you a bit what is the effect of, what is the effect of EEs on microtubule dynamics, right?
Yeah, the functional aspects I will discuss it in the next lecture.
Here I just give you now the basis how the proteins come together and how you can now tweak the system or manipulate the system.
So can you look for these domains because protein is also a those unknown proteins?
I will discuss this as well.
Yes.
I will show you.
Very good point. Yes, I forgot to mention this. Actually, it's true. So based on this concept, we were able to use this to search down the entire genome. And Anna will tell you more about that. So indeed, it has predictive power, I showed you. Yes?
How is the plus-tip complex composition changes during catastrophe?
So you lose all plus tips when the microtubule it's not completely true. I would say the majority of plus tips you lose them as soon as they start shrinking, because the EEs, they don't track depolymerizing microtubules. However, there are a few examples of proteins, and maybe Anna will also show you one, that can track also shrinking microtubules in an EB-independent manner. Yeah, good question.
One of the previous proteins which was not naturally localized in the tips of the microtubule, once you introduce this domain, so it was able to localize in the tips of the microtubule. What was the effect on the cellular phenotype?
Yeah, that's also a very good question. I think we did not look too much into this. So this question is why the KIF2A, whether the KIF2A by localizing it.
I couldn't hear it. Can you repeat?
Okay, so his question was, he was referring, oops, that's the wrong way. So he was referring that, so we have here a protein that is not a plus tip. You introduce the SxIP motif, you localize the growing microtubule, what's the phenotype? Right, and I think we did, that's a very good question. I think we haven't looked too much into the detail what is going on here.
I will touch upon it, okay?
Okay, good. Even better.
So,
Yes?
is there any domain which is also known to recruit some of the functional protein at the tips
I think I didn't understand the question. Where?
Is there any domain which can recruit other proteins, functional protein at the tip?
So you mean if we have recruited an SxIP protein, whether now additional protein can bind to the SFA? Yes. So there is a hierarchy between the plus-tip interactome. So the lowest hierarchy we think are the EBs, or the highest hierarchy, I mean the first level of hierarchy. Then there is the second one that is mediated by these SxIP proteins or CAP-Gly-mediated, I just come to that. And then there are additional levels.
Is it different from this domain? This is only...
Now I show you another example right now, that there are other domains involved. Yes?
Maybe I will check for the size how what is the ratio of EB and like 3D microtubule inside? How much EB per 3D microtubule? 3D tubule.
Yes. So Anna will show you that there are roughly 700 GTP tubule. The GTP cap is more or less 700 tubulin long. So we think there are hundreds of EBs localized into a growing microtubule tip. And on top of that, all the others as well. So it's going to be very crowded there.
And what about 3D, which are not localized in microtubule? 3D EB or...
I guess they swim in the cycle, so. Yes?
So many SxIP domain containing proteins can bind to the EBs. But then how is that regulation happening? As all of them can bind, right? Integratively.
Very good question, we have no clue. So that, yes, Anna.
Sure. I do have an answer to that one, because the concentrations of the proteins have been now determined by using mass spectrometry. So there are a couple of mass spec papers where they have counted the number of proteins per cell for abundant proteins. And one thing that you find is that EBs are approximately 10 times as abundant as almost any other protein which can bind to them, 10 or 100 or maybe even 1,000-fold more abundant, depending on the protein. So the way we think about it, that the EBs are actually very abundant, so they decorate the tip, and all the partners will also bind them, but since they are less abundant, normally they can compete with each other to some extent, but there is still space for everyone.
Is it like some sort of a domain organization where a particular type would be in a maybe particular cell type or in a particular...
So there are also cell type dependent proteins, regulation dependent proteins, and also proteins that are located to specific places in the cell. And I've also shown it.
Would you like to comment why so many proteins would be required to bind to the EBs?
Let's discuss it at the end, okay? Let's just go push on. Actually, first get a full picture of the structures. And then I will tell you something about what we know, about the function, and then in the end we can all together think why there are so many, okay?
Okay, so in principle we can, with this one complex structure I showed you, already explain this part of the plus-tip interactome, so the largest part. So here I have divided between SxIP proteins and other proteins, so or at least half of it we can already explain just by this one complex structure. Now, interestingly, besides SxIP proteins, we have also two very prominent examples of plus-tip that carry a so-called cytoskeleton-associated protein glycine-rich domain, or abbreviated CAP-Gly. I've already touched a bit on them, and that's another group of proteins we have looked at in quite large details. So what are CAP-Gly-domains? CAP-Gly-domains are small protein modules, roughly 80 amino acids in length, that are glycine-rich. And the glycines, you can immediately, hopefully, recognize those on the back. They are highlighted here in red. They are highly conserved, and they are involved in shaping the loop regions of this otherwise beta-sheet-rich globular domain. And they are highly conserved, again, from yeast to human. The most conserved region of CAP-Gly-domains is this part here between the beta-3 and beta-4 loop strands, and it harbors the so-called GKNDG motif. You can recognize it here in the middle, and I will also come later what is the relevance of this GKNDG motif, so just keep it in mind. Now, where do CAP-Gly proteins appear in cells? So one prominent members are involved in tubulin biogenesis. So the tubulin binding cofactor A and B, especially the B has a CAP-Gly-domain. They are involved in folding the alpha and beta tubulin subunit and assembling the heterodimer. So I don't know whether we have heard so there's quite a complex machinery just to produce this alpha-beta tubule heterodimer. And that's also probably the reason why so far people failed to produce, for instance, in E. coli, but also well, primarily in bacterial systems, because if you want to overexpress tubulin in human cells, you obviously end up with some problems. But they definitely fail to produce recombinant protein. And the reason I think is because there is quite a complex machinery involved in assembling this heterodimer. There is a kinesin, KIT13B, that harbors a CAP-Gly-domain. There are molecules at the Golgi that have CAP-Gly-domain. At the centrosome, CAP-350, for instance, is a good example. And then we have, as I just mentioned, also at the positive P150, the dynactin, large dynactin subunit P150 glued, and the clip 170, and a shorter version, clip 150. So I will focus mostly on those. Again, the work I will present, I took them from these papers again. Most all of them were done in collaboration with Anna. Srinivas, prominently represented in these two papers who did the structural work. Then a PhD student in Anna's lab, Babette, that is now in Vienna as a postdoc, did a lot of work on the cell biology part of this molecule here, and Anke, PhD student in my lab. So, when we looked now at these CAP-Gly proteins, we first have to assemble a little network so that we are able to study them. And we selected these 4 proteins. So we have again here the EBs, with the EB homology domain here, this time now just schematized. We have SLAIM. Anna will talk more about SLAIM also in her next talk. Then clip with the CAP-Gly domains of clip 170 and the CAP-Gly domain of P150 group. And the first interaction we've looked at, which was already documented in the literature, was the interaction between the CAP-Gly domain of P150 group with the C-terminal domain of EB. So we succeeded to produce a crystal structure between, again, the C-terminal domain of EB and the CAP-Gly domain of P150 group. So here's the C-terminal domain of EBs, and here the CAP-Gly domain of P150 group. And what immediately popped up is that we are looking at the bipartite interaction mode. So you see 2 different interaction sites. The first is between residues in the 4 helix particle of the EB, so this part here, with the blue residues on the CAP-Gly domains here, and another interaction between a hydrophobic cavity here highlighted in green with the last 3 C-terminal residues of the EB. Remember, I told you that EBs have an EEY motif at the very C-terminal that is highly conserved. So a bipartite interaction. So I don't want to go too much into detail, but I want to highlight what we see here at a bit high resolution. So what we saw is that the very C-terminus of EB, here in yellow, packs against the beta-6 strand of the CAP-Gly domain. And what we revealed in the structure, or saw in the structure, is that this terminal tyrosine residue here, together with the carboxylate group that is free because it's the very C-terminus, they are recognized by an asparagine, by a lysine that stems from this GK-MBG motif I just told you earlier. So very interesting, these 2 residues, highly conserved amongst CAP-Gly domain, they recognize these C-terminal tyrosine residue. The other thing we also saw is that the ring here, the hydrophobic ring of the tyrosine, packs against hydrophobic residues on the CAP-Gly domain. So it turned out that this GK-MBG motif is apparently involved in recognizing C-terminal tyrosine residues. Now, to get an idea whether this is something conserved in CAP-Gly domains, this interaction mode, the next structure we solved is between the CAP-Gly domain of P150 group with the C-terminus of CLIP, which, when you look at the sequence alignment of the C-terminus, has not a tyrosine but a phenyl alanine, so a very similar residue, just lacks one OH group. And the C-terminus of CLIP also has a zinc knuckle domain, so it's a zinc binding domain. So what we did, we solved the structure between the CAP-Gly-domain of P150 GLUT, and the C-terminus encompassing the symptom and the EEY motif of CLIP. And again, what we saw is a bipartite interaction mode, as we have seen it with EE1C and P150. So here, as in surface representation, is again the CAP-Gly-domain of P150, and in yellow, the C-terminus of CLIP. Here's the zinc couple, it's a CCHC zinc couple, so there are 3 cysteine and 1 histidine that are involved in coordinating the zinc atom. And we again see, in this case, the ETF motif of CLIP binding into this hydrophobic cavity on the CAP-Gly-domain, and we found basically the same binding mode. So again, the asparagine and the lysine of the GCAM-DG motif implicated in specificity recognizing this terminal phenylalanine. The last structure we looked at, complex structure, is this time not between the CAP-Gly domain of P150, but between the CAP-Gly, the first CAP-Gly domain of CLIP170 and the C-terminus of SLAME, predicted to be disordered. And what was the first surprise when we saw that there is indeed a binding, basically unaccounted desire, is that while the tyrosine is there, the C-terminal tyrosine is there, the sequence homology to the other candidates is much weaker. But nevertheless, we saw good binding. So we saw the structure and found again a bipartite interaction mode, again with the tyrosine that is coordinated by the GCAM-DG motif. But on top of that, we saw that a tryptophan residue explores a cavity on the CAP-Gly domain of CLIP. So do you see this here, this tryptophan that is only present, this cavity is only present on the CAP-Gly domain of CLIP and not P150. So we did not see binding of this peptide to P150, but only on CLIP, so telling us also the specificity. But important for right now is again we saw a bipartite interaction mode. Now when we superimposed a few of our complex structure, we saw that the binding of these terminal tyrosine is really identical. So these already, with this information, we were able to define that actually CAP-Gly domains are C-terminal EEY motif recognition domains. CAP-Gly domains are made to recognize C-terminal tyrosine residue, and it's the GCAM-DG motif that is responsible for that. But on top of that, I always told you there is always bipartite interaction mode. So besides this EEY binding, there are other places on this very small domain, by the way, that are exploited to make additional contacts, either with the EB homology domain, in the case of P150 GLUT, or with the zinc knuckle domain of CLIP170, or this tryptophan residue we have seen in SLAME. So it turned out that these CAP-Gly domain are multi-purpose platforms to mediate diverse positive interaction. And when now you figure out what kind of interaction can happen, you immediately see it can form quite complex interaction networks. So a little domain again, a little motif, adding quite a lot of complexity to the system. So similar to the SxIP situation, we also wanted to know whether what we have seen in our crystal structure is functionally relevant. And again, by testing this in cells. And I want to show you now one example of one experiment we did in cells. Basically, Anna did the experiments, if I remember correctly. Was that, so we used a construct of CLIP. So CLIP has two CAP-Gly domains, the coil domains, and this EEY motif at the very C-terminus. We fused a YFP tag to the N-terminus. We translated that into cells and overexpressed it. And when you do that, you don't get only localization to the growing end, but you start to decorate the entire microtubule cytoskeleton. And this is a typical phenotype when you overexpress too much of a plastic, you get also lattice binding. But nevertheless, we wanted to use this situation to ask the question whether this overexpressed CLIP all over the microtubule cytoskeleton is able to relocalize the P150 GLUT, endogenous P150 GLUT, to the CLIP-labeled microtubule cytoskeleton. And that's what you see here. This is the situation where Anna used an antibody against endogenous P150 group, and it very nicely co-localized with the overexpressed clip-decorated microtubule cytoskeleton. The next experiment we did is then, since from our structure it was clear that we need a free tyrosine, it has to be present at the C-terminus, we fused a CFP at the very C-terminus to block access to this tyrosine. And what happens? You lose completely relocalization, so consistent with the structural data. Then we moved the CFP from the C-terminus to the N-terminus by freeing the phenylalanine in this case, we got relocalization. And finally, we just chopped off the EEY motif and there was no relocalization. I think this is quite an impressive, easy but impressive experiment, because when you consider that P150 glued is part of the dynactin complex, which constitutes of more than 20 subunits, it's a megadalton complex. On top of that, it is a cofactor of cytoplasmic-dynein, which by itself is a megadalton complex. It turned out that by this very minor interaction, you can relocalize this entire system where you want to have it. So, when you look at the possible interactions between CLIP, P150 glued, and CLIP can also undergo an autoinhibition, self-associated head-to-tail association. I did not went into the details. It turned out that all these interactions that are highlighted here, the structural basis to explain them is this little interaction mode between the CAP-Gly-domain and the EEY motif. Now, these data also have further implications, and I will just touch, I think, on 2 or 3 of them. The first is this CAP-Gly-EEY recognition useful also to target CAP-Gly-domains to growing microtubule? So remember, EVs are autonomous tip tracking molecules. They localize to the growing microtubule end. They bring each of them, because they are dimers, 2 EEY motifs. And on top of that, as you probably all know, alpha-tubulin also has an EEY motif. So the concentration of EEY motifs increases dramatically at the tip, at the growing tip. So we suggested at that time that indeed it's the increase in local concentration of these EEY motifs, together probably with secondary binding sites on the microtubule, which were described to exist, that they drive the localization of CAP-Gly proteins to the growing microtubule. So it's a different mechanism what SxIP proteins use, but the end effect is exactly the same. Then the data have also implications for the tubulin tyrosination cycle. We also heard from Peter Bass about tubulin tyrosination. And just to recapitulate a bit what you already heard, so it's very well known, actually since the 70s, that the alpha-tubulin undergoes a detyrosination cycle, where tubulin tyrosine carboxylate peptidases can remove the terminal tyrosine to produce so-called blue tubulin, because there is a glutamic acid at the very C-terminus left after this reaction. And there is a ligase, tubulin tyrosine ligase, that can add back this tyrosine to form again tyrosinated tubulin. There is also a second reaction going on here where an so far unknown enzymes removes the terminal glutamic acid of the detyrosinated alpha tubulin to produce so-called delta-2 tubulin, and that cannot be rescued back. So, what is the functional role of these tubulin cycles? Although we know it since the 70s, rather recently we are starting to understand also its functional role. And actually what turned out is that if you suppress the tyrosination cycle, this leads to defects in spindle positioning, in abnormal cell morphology, also in disorganized neural networks, and actually it leads also, and that has been shown by a knockout mouse, to perinatal death in mice. So these mice, they don't survive, they get born and they live for less than 1 day and die. So it's an absolutely essential enzyme. And people from Didier Job and others, they then found that this tubulin tyrosine is also a major factor that affects the recruitment of CAP-Gly proteins at microtubule ends. And I think, again, it's very easy to provide insights why that is, because, as I told you, this EEY motif is the motif that is recognized by the CAP-Gly domain using the GKNDG motif. So this tyrosine is absolutely essential. And if you don't have it, in principle, you cannot localize CAP-Gly domains to growing microtubule. So one molecular function of this tyrosine is to recruit CAP-Gly proteins. And that could be one part of the story why these mice die after birth. So I come to the end. So what I have shown you is that there is this part of the plus-tip interactome that use SxIP motifs to localize to the EE homology domain, and I showed you that CAP-Gly proteins, these two prominent examples, they target the EEY motif, and that by just solving two complex structure, we were able to cover almost 80% of the entire, to understand on a structural basis 80% of the plus-tip interactome. Are there questions concerning this topic?
I have a question that in EB1 we say that EB1 includes other plus and minus proteins to the plus and of the minus proteins. So does it bind to those proteins in the cytosol and then brings it to the tips, or its presence itself at the tips makes others to come there and
What do you think on that? I think rather they bind to the tip and recruit others there. They don't drag them there.
The other proteins, they somehow come to know that the EEY is present at the plus and which also
Yeah, so I would say that the second option, that the EB proteins have first to bind to the tips and then recruit their partners to the tip, is the one which normally occurs. And one reason to think so is what Michel just showed, is that many of these proteins also have their affinity for the microtubule. So like, for example, for CLIP-170, it actually has 4 of these CAP-Gly domains, and has been shown in vitro that you need interaction with both. So you will have a high concentration of affinity sites both for EBs and for microtubules. Also, you have a high concentration of EBs there, so that's another factor. And there's also been ideas that EBs might be actually self-inactivating as well, so that when we bind to microtubules, that then somehow the binding to the partners will be more accessible, although we're not sure that it's true. I should say that when you overexpress any of these proteins, you will find interactions in the cytosol, and you will also be able to recruit EBs to other sites where some of these partners will be residing. So let's say some of these proteins bind to the membrane of the kinetochore, and if you elevate the concentration of EBs or other proteins, it will go there. But normally, EBs are just cytosolic and at the tips. They are not very prominent elsewhere, which also tells you in a way that probably it's somehow regulated in such a way that EBs really bind their partners mostly when they are at the tip. How this works, we don't really understand.
And if it has an affinity for the microtubule also, why can't it directly go and bind to the end without the EB1?
Yeah, so most of them also have some affinity for the microtubule lattice. Part of it, I think, also as Michel very nicely showed you, is that there is also an electrostatic component to the interaction. And many of these proteins, they would have these positive charges. The lattice of the microtubule is very negatively charged. That's one of the most important properties of the microtubule. So there will be also electrostatic kind of crowding interaction.
So is there an interaction between GTP and EBs? Because
We will hear this afternoon during the discussion of the paper. Yes, there is. I can tell you already. And you will see how that's done. Yes?
Is there an active analog of any of this?
What?
Is there an active analog of any of interaction?
Whether growing actin filaments also have plus tips, let's say. Formins, the formins, yes. Yes, true, the formins. Yes?
I got the impression that there is only few amino acids necessary for the interactions, right? These motifs are very small.
Yes, they are. With these two, you can already explain a lot. Yes.
Okay, so question is how they why there is a possibility of non-specific interaction, right? If they are like only these domains are very small. So how they are?
Yes. Yes, I understand. I hope I have understood. So, of course, these are the conserved interactions. So here, as just Anna also mentioned, you need also electrostatic interaction. Unspecific, long range, but they help to make the system more robust. In the case of the CAP-Gly-domain, I showed you that it's always a bipartite. In that sense, that's also a bipartite interaction mode. You have specificity, hydrophobic interaction, hydrogen bonding network, which mediates specificity and an electrostatic component. So bipartite, and you need both, basically. And here, there is, I showed you that there are EB homology domains that combine on this side. So it's also a bipartite interaction mode. And depending whether the features, so these are the conserved features, depending on the feature, the additional features that are encoded under the CAP-Gly-domain, you can distinguish between whether the P150 CAP-Gly-domain should bind the EB homology domain, and the CLIP-170 domain has another specificity, for instance, for this tyrosine of Slane. So you can encode onto the surface of the CAP-Gly-domain's additional specificity determinants. So these are the ones that are conserved. And the rest is specific depending on which CAP-Gly-domain you look at. Okay, so in the end I just want to have 2 or 3 slides showing you a bit our institute in Switzerland. So that's the Klaus Sterer Institute. It's situated between Zurich and Basel. And it's a physics institute, so you may wonder why, as a biologist, as a protein chemist basically, with structural biology, of course, emphasis is working at that place. The reason for that is that, so that's our labs. This is the place where we have robots to crystallize our proteins. And what we do then to measure the crystals is that we walk over this bridge here, and to reach this round building, that's the view by night. That's the Swiss light source, a synchrotron, a very powerful machine for really doing excellent crystallography work. And we have also special type of equipment there is that, so for those that know how crystallization is done, you crystallize your protein in these 96 well planes. And typically, when you have a home source or when you go to other synchrotrons, you have to fish them out. That's a bit tedious, you have to freeze them. So we have an equipment where we can directly stick the plate into the beam to screen for diffraction. And that's exactly the information, the type of information you want to have. You want to know whether the crystal that looks beautifully, and I can tell you often when they look beautifully, they don't diffract very well. And here, you cannot just look them optically, but directly on the information you're looking for, namely on their power to diffract X-rays. So we can directly screen in situ the quality, the diffraction quality of our crystals, and that helps enormously to speed up the process of optimizing the crystal. Because based on this information, when they go back, well, either we can then fish them as well, we can also fish them and then do a data collection, but if they are too small, we can go back to the lab, try to get and improve this condition, try to get a better crystal, go back to the beamline and back and forth. And since this is a little distance between the lab and the wood machine, we are quite fast also in producing structures and makes us competitive. And with this, I want to thank you for your attention. And I guess we have a tea break, right?
Yes, we're doing a tea break now.